stage 3 biology. Actually, this is the first checkpoint biology material I'm doing for my year 8 student, sorry, because the exam is fast approaching and most of the students ask me to do a video for them, which they will use to revise their content before the exams for the autumn term. So quickly, we'll be looking at the learning objective we'll be looking at for this lesson is to describe the stages of digestion in humans. We should be able to identify part of the alimentary canal and their functions. We should state the types of digestion. We should also describe mechanical digestion, where we identify the types of teeth and their functions. We also describe the causes of tooth decay and possible method of prevention. So we are going to also identify the enzymes that are responsible in the process of chemical digestion. So here we look at the stages of food breakdown. Now, the food taken into the body goes through 6 major stages during its passage through the alimentary canal. Now, these 6 stages that food goes through when it passes through the alimentary canal is ingestion. Ingestion means the taking of substance into the body when through the mouth, and things like food and drinks that you take into your mouth is the process of ingestion. Now, once the food gets into your mouth, the next stage that we follow up is mechanical digestion, which is the breakdown of food into smaller pieces without the chemical change to the food molecules. That means if the food that we take in, be it carbohydrate, lipids, proteins, are all insoluble. So you find that mechanical digestion does not break the bond that hold the molecules together, but however, it breaks it down to smaller pieces to increase the surface area so that the process of enzyme and chemical digestion can take place easily. Then we have chemical digestion also, which has to do with the breakdown of large insoluble molecules into small soluble molecules. So the difference between mechanical and chemical is in chemical digestion there is a change in the chemical content of the molecule of the food molecules, whereby the food itself will be broken down. It will be converted from an insoluble state to a soluble state. Now the major reason is the food cannot be absorbed if it is insoluble, so it will be difficult for it to diffuse and be absorbed through the alimentary canal, because the alimentary canal is selectively permeable. But however, once it is soluble, it will be easy for it to move down the alimentary canal into the bloodstream. The next is absorption. Absorption simply means the movement of small food molecules and ion through the wall of the small intestine into the blood. So I need you to take note of this. In absorption is the movement of these food molecules and ion through the wall of the small intestine into the blood. It is absorbed into the blood. Then in assimilation now is the movement of these digested food molecules into the cells. So it moves into the cells of the body where they are used and become part of the cell. That's the difference between absorption and assimilation. So in assimilation is the movement of these food molecules from the blood into cells where they will now finally be used and becoming part of the cell. Then ingestion is the passing out of food that has not been digested. So the removal of undigested food, which is in the form of feces through the anus, is known as ingestion. So these are the major stages of digestion in humans. So, however, we'll now look at the part of the alimentary canal and their functions. So, part of the alimentary canal or the gut and their functions. So, the first thing is the mouth. Now, where food is being, you chew the food and mix with saliva in the mouth. Now, so the chewing action here is mechanical digestion. However, the saliva contains an enzyme called amylase. Now, this enzyme called amylase help in the digestion of starch. So you find out that the process of chemical digestion of starch starts in the mouth also, and mechanical digestion by the action of the teeth also start in the mouth. Now, here you have your salivary gland, which releases saliva that help in the digestion process. Now, once the food is being mixed with saliva and broken down into smaller pieces by mechanical digestion, then it forms bolus, and the bolus will now move down through the esophagus. Now, here you have the esophagus here. esophagus. Now, which moves the food to the stomach. So the esophagus connects the mouth to the stomach, and the food moves through the alimentary canal through a process called peristalsis. The food moves through the alimentary canal through a process called peristalsis that involves two types of muscles, which are the longitudinal and circular muscles. Then from here, the food moves to the stomach, to the stomach, and at the stomach, sorry, what happens is it mixes and churns the food with gastric juices. So mechanical digestion takes place too in the stomach through the churning action of the stomach, and it mixes with gastric juice, and this juice contain an acid and a protein digestive enzyme. So it contains hydrochloric acid, which has a pH scale of approximately 2 to 3, which means it is a concentrated acid. Um, we'll look at the function of the acid much later, but it also contains a protein digestive enzyme called pepsin, which also starts the process of protein digestion. Then the food now leaves there and move to the small intestine. Now, in the small intestine, what happens is the digested food are absorbed. The digested food and absorb nutrients into the blood or the lymph. Now, fat and oil are absorbed into the lymph, that's fatty acid and glycerol, while glucose and amino acid are both absorbed into the blood stream at the small intestine. Now, you also have the large intestine, which absorb water and some minerals and vitamins. Um, home and is also home to intestinal bacteria and passes waste product. Then the anus open to allow waste to go out. Then you also have the rectum, which help to store waste. Now, these are the major organs, but however, we have certain accessory organs that also help in the process of digestion, and one of that is the liver. Now, the liver is used to make bile, which aid in the digestion and absorption of fat molecules. And it also helps to provide the optimum pH for enzymes within the upper part of the small intestine to work. Then you also have a gallbladder. The gallbladder major function, it helps to store bile. So you should know the bile is produced in the liver and stored in the gallbladder. Then you have a pancreas, which release bicarbonate to neutralize the intestinal content. Remember, the intestinal content is acidic when it leaves the stomach, so it helps to neutralize it, and it also produces enzymes that help in the digestion of carbohydrates, protein, and fat. So you need to know the part of the alimentary canal and their functions. Very, very important during checkpoint biology exams. Then next, there are two major types of digestion. We have the mechanical digestion and the chemical digestion. So we'll look at the mechanical and chemical digestion now individually. So mechanical digestion, what does it do? Now, where does mechanical digestion happens in humans? You need to know that. Now, mechanical digestion is the breakdown of food into smaller pieces without chemical change to the food molecules. I've said that earlier when describing the stages of food digestion. Now, it is mainly carried out by first the chewing action of the teeth when you use your teeth to chew the food, and secondly, we have the churning action of the stomach, and lastly, the emulsification of fat by bile. So three paths in the alimentary canal where mechanical digestion take place. First, the chewing action of the teeth, and the churning action of your stomach, and the emulsification of fat by bile, which take place in the duodenum, which is the upper part of the small intestine. So next, importance of mechanical digestion. Now, one is it helps to in the break up. It helps to break up food into smaller pieces. That's one function. It helps to break up food into smaller pieces. Two, it helps to it increases the surface area for enzyme action, and it also make it easy for you to swallow the food when it forms bolus and through the alimentary canal. So now let's look at the teeth. You find that our teeth are held firmly in the bone of the jaw. So the different shape The difference in shape and sizes of the teeth enable them to perform slightly different functions. So there are four major types of teeth, um, which is the incisors, the canines, the premolars, and molars. Now, the incisors are usually chisel shape for biting and cutting of food, and the canine are usually pointed for tearing, holding, and biting of food. And you have your premolars and molars, which have larger flat surface, usually used for chewing and grinding of food to increase their surface area and break it down to smaller pieces without changing their chemical content. Now, if you look at the tooth here, you find in front of your jaw, you find the incisors, which is used for cutting and biting. Then immediately after the incisors, you have your canines for holding and tear and cutting also. Then your premolars and molars, which are used for chewing and crushing of food. Now, I think this will give you a broad, clearer picture of how it works. So you find that this whole four here forms your incisors. Then you have your canine, then you have your premolars, then three molars here. Now you find out that the incisors and canine only have a single root, while premolars and molars have two double roots and quite a flat, large surface. Now, tooth decay and tooth care. What are the causes of tooth decay? Why will there be a tooth decay? Tooth decay is caused by bacteria. So it further that bacteria produce a sticky matrix when mixed with saliva. Now, if there is bacteria in your mouth, it will now form a sticky matrix when you mix it with saliva, and the saliva will now trap food. The saliva matrix with bacteria traps food, which will form a coating of plaque in the mouth. Now, the plaque help to convert sugar to acid. So the plaque will now react with the leftover food to produce an acid. Now, through a process called anaerobic respiration. Now, the acid will now convert or remove the calcium that the teeth is made up of, thereby dissolving the dentine and enamel, which are the two major part of the teeth that gives it a strong and firm nature. Now, what are method of treatment of tooth decay? You find out that decayed part is drilled out by dentist, and the hole is filled with amalgam, which is an alloy of mercury and another metal or plastic. That's the major method of treatment. But however, there are ways in which you can prevent tooth decay. Now, method of preventing tooth decay is to reduce the eating of sugar content. Another way is regular and effective brushing of your teeth at least twice a day to prevent buildup of plaque. The third one here is you finish a meal with a crisp vegetable or fruit and rinse with water effectively. Then regular visit to a dentist to ensure that any sign of gum disease or tooth decay can be dealt with properly. Then you need fluoride should be added to drinking water to reduce the risk of dental decay. So that's also very important in preventing tooth decay. Now let's look at chemical digestion quickly. That's it about mechanical digestion. So chemical digestion is with the aid of enzymes. So you find out that enzymes are usually biological catalysts that help to speed up the rate of chemical reactions in living organisms. Now, without enzymes, you find that reactions in living organisms will occur too slowly, that it might not even be enough to sustain life. So you find out that the purpose of digestion is to break down, of chemical digestion is to break down large insoluble molecules into small soluble molecules. Now, these insoluble molecules include your carbohydrate, protein, and lipids. Remember, lipids are your fat and oil. Fat is solid at room temperature, while oil is liquid at room temperature. Now, once it is converted to small soluble molecules so that they can easily be absorbed into the bloodstream. Now, food is partially digested by mechanical process, which we've looked at, by the chewing, churning, and emulsification process. Chewing by your mouth, churning stomach, emulsification by fat in the upper part of the small intestine called the duodenum. Now, in order to break large pieces of food into small pieces of food, which increases the surface area for enzyme to work on. So you look at this, you find that digestion mainly takes place chemically, where bonds holding the large molecules together are broken to make smaller and smaller molecules. Now, chemical digestion is controlled by enzymes. Chemical digestion is controlled by enzymes which are produced in different areas of the digestive system. Now, there are three major types of digestive enzyme you need to know for key stage 3, and they are the carbohydrate enzyme that break down carbohydrates to glucose, the proteases enzyme that break down protein to amino acid, and the lipases enzyme that breaks down lipids to fatty acid and glycerol. So quickly, let's just look at them individually. Carbohydrates are digested in the mouth, stomach, and small intestine. Those are the three places where carbohydrate is digested. Now, carbohydrates enzyme break down starch into sugar, simple sugar such as glucose. Now, you find that there are two types. We have the amylase that help to break down carbohydrate to maltase, and we have maltose that break down to maltose, and we have maltase enzyme that break down maltose to glucose. But you don't really need to do that much detail for checkpoint. You just need to know about carbohydrates enzyme breakdown carbohydrate to glucose. So you find out that the saliva in your mouth contains amylase, which is a type of carbohydrate enzyme, which is another starch digestive enzyme. So if you chew a piece of bread for a long enough time, you find out that the starch it contains will start to be digested into a small sugar or glucose. Thereby, it becomes more tasty, it becomes sweeter the longer you chew it. So you find out that this is more like a carbohydrate molecule. You find out the carbohydrates are bond together. So it's a group of these glucose molecules that join together to form a large starch molecule. So the function of the carbohydrate enzyme is to break this bond that is holding the glucose molecules together. Now, once the bond have been broken, then you now have the smaller soluble molecules called glucose. So next is protein. So protein are digested in the stomach by the enzyme called pepsin. I've stated that earlier, and the small intestine. So proteases enzyme break down protein into amino acid. Proteases enzyme break down protein into amino acid. So you find out that digestion of protein in the stomach is helped or assisted by the stomach acid, which is a strong hydrochloric acid. A strong hydrochloric acid. Now, the function of the acid is this acid also kill harmful microorganisms that can be in the food. So here is simple. So you see the bond holding the protein molecules together. Now, if you study our video on healthy diet and malnutrition, I explain this bond in detail. So the protease enzyme will now help to break this bond holding the molecules together to now form amino acid through this process of digestion. So the enzyme for digestion of protein is protease enzyme. While for fats, remember I told you is the lipases enzyme. Now remember I just used fat here. It's supposed to be lipids, and there are two types, which is fat and oil. Fat is solid at room temperature, while lipids is liquid at room temperature. So you find out that the lipase enzyme break down fat into fatty acid and glycerol. So fatty acid and glycerol. While carbohydrate is glucose, protein is amino acid, while lipids is fatty acid and glycerol. So you find out that digestion of fat in the small intestine is helped by bile. Digestion of fat in the small intestine is helped by bile, which is made in the liver and stored in the gallbladder. So bile break down fat into small droplets that are easy for the lipase enzyme to work on. So you find out that these are fat molecules, and the lipase enzyme will now break this bond here called ester bond that hold these molecules together to form fatty acid and glycerol. That's it. So we'll just look at the role of hydrochloric acid now. Remember I told you that hydrochloric acid is in the stomach. And what is the function? First, the stomach produces several fluids, which together are known as gastric juices. But one of the fluids produced is hydrochloric acid. And the function of the hydrochloric acid is it helps to kill bacteria in the food and give an acid pH for enzyme in the stomach to work, majorly the pepsin enzyme. That's the two major functions of hydrochloric acid. Then next, we'll look at the role of bile. Remember, I told you that bile is produced in the liver and is stored in the gallbladder. Now, the bile is being released from the gallbladder into the upper part of the small intestine called the duodenum here. And the major function it helps to do is this. So bile, it is alkaline, so to neutralize the hydrochloric acid which comes from the stomach. And two, the enzyme in the small intestine have a higher, that means it has a more alkaline optimum pH than those in the stomach. So you find that another function of bile is it breaks down large droplet of fat into smaller ones, and this is known as emulsification of fat. Thereby it helps to increase the surface area to allow lipase enzyme to work on in order to break down these lipids to fatty acid and glycerol faster. Then how is food absorbed into the blood? Simple. So absorption of food and water. So absorption is the movement of digested food molecules from the digestive system into the blood. So glucose and amino acid is absorbed into the blood, while fatty acid and glycerol is absorbed into lymphs. So we have the lymphs here. So water is absorbed in both the small intestine and the colon, which is the large intestine. But most absorption of water happens in the small intestine. So reabsorption of water takes place in the large intestine. However, absorption takes place in the second section of the small intestine, which is called the ilium. So absorption takes place in the small intestine part, which is called the ilium. So if you remember ilium for absorption, good and fine. If you remember small intestine, you still get your mark for absorption of food. This is key stage 3. If we're looking at IGCSE biology, then you have to be extremely specific. Now, how is the ilium adapted for absorption? It's simple. Sorry about that. You find out that ilium is adapted for absorption as it is very long and has a highly folded surface with millions of tiny finger-like projection called villi. Now, it is very long and have a highly folded surface with millions of villi, which are tiny like projections. Now, I find out that these adaptations massively increase the surface area of the ilium thereby allowing absorption to take place faster and more efficiently. And you find out that it also have high concentration of blood capillaries and also lacteal. So the food can easily be absorbed into the bloodstream, the glucose and amino acid, while the fatty acid and glycerol can now be absorbed into the lymphatic system or the lacteal. Now, so this is just the structure of it, of the ilium. You see, it has a large folded surface, which help to increase the surface area to make it easy for food as absorption. So, thank you so much. So maybe we'll be looking at other aspect of key stage 3 biology based on request. So if there is a high request of me doing it, then I think I might just have a go of it. So have a nice day. Thank you.